Hello students, welcome to e-learning classes. I am Piali Nag, your biology teacher, and I will be taking your next chapters of the biology. Okay, so let's get started. We will start with genetic code. In my last video, we have already gone through transcription in prokaryotes as well as in eukaryotes. Before that, we have gone through the characteristic features of DNA and DNA as a genetic material and replication of DNA. After we finish the second portion of the central dogma, that is transcription, and the first portion was DNA replication, we start with the last part of central dogma, that is translation. Before we go through the translation, we need to know what is a genetic code and what are the salient features of genetic code, the tRNA molecule, and so on. So first we will go through the genetic code. What is a genetic code? It is actually a relationship between the nucleotides on the mRNA, which was being transcribed from the template DNA and was modified, containing only exons, and the sequence of amino acid in the polypeptide which is going to be formed, that is called the genetic code. Now it was suggested that the code must consist of 3 bases to code for 20 amino acids, as there are 20 amino acids known till date, but now it is 21 amino acid. Okay, so but till then we knew that there were 20 amino acid in nature. Okay, so and there are 4 bases, that means 4 cube, that means 4 into 4 into 4 becomes 64 base pairs which code for 20 amino acid. So codon is actually a triplet. Triplet means it is made up of 3 base pairs. Okay, later this genetic code was developed by Hargobind Khorana, where a chemical method for synthesizing the RNA molecules with the defined base combinations like the homopolymers and the copolymers were used to develop the genetic code. Okay, now what are the basic before going to the salient features of the genetic code, we also need to know that the genetic code was artificially synthesized proteins. To understand, actually the proteins were artificially synthesized to actually understand the nature of codons. Okay, and few scientists also demonstrated that the polynucleotide phosphorylase also helped in polymerizing the RNA with particular sequences in a template independent manner. Okay, which is an enzymatic RNA synthesis. Now coming to the basic features of the genetic code. Genetic code, if you go through the diagram, that the codons are actually triplet in nature. That means one codon is made up of 3 base pairs. Now out of 64 codons, only 61 code for 20 amino acids. Then where will the last 3 go? Actually the 3 codons, particularly UAA, UGA and UAG, do not code for any kind of amino acid. They does not actually have or express any property of any amino acid. So they are called as stop or terminating codons. Okay. Next is, one codon will code for only one particular amino acid. So the code is unambiguous and very specific. That means if you go through the diagram, the first one is triple U, and which codes for phenylalanine, and triple U will always express the property of phenylalanine amino acid. So it is very specific, but some amino acids are coded by more than one codon. Hence, the genetic code is also called degenerate. Okay. Now, if you go through, triple U was always phenylalanine. Okay. But UUC is also phenylalanine. That means triple U will always code for phenylalanine, and UUC will also code for phenylalanine. That means this particular amino acid can be coded with these two genetic codes. So the codons are specific. The codons can only code for only one amino acid, but that particular amino acid can also be coded for another base pair, a combination of another base pair itself. Okay, the codons which is read in the mRNA is in a contiguous fashion. That means it does not contains any punctuation. Okay, and so it is, the code is actually commaless, and the genetic code is nearly universal. That means this particular genetic code is accepted throughout the world, and it is same for almost 99% in all the organisms except in mitochondria and few protozoa. Okay, now, and if you find that the AUG codon, it has two functions. Okay, that it is first termed as the start codon, because whenever there is a AUG codon, the process of transcription actually starts from there, and it codes for the amino acid methionine. So it acts as an initiator codon, and it also codes for the amino acid methionine. Okay, now coming to the mutations as well as the genetic code. Now, what is actually mutation? You have already gone through in chapter 5. So mutation, again I will not describe, just I'll go through, I'll give you an overview. That is, mutation is actually a sudden inheritable change in the genetic material. Okay, now mutation, every generation causes mutation. Okay, mutation always, a change always does not means a disease. If that means we all are changing, like we all are different, dissimilar from our parents, right? Genetically we all are dissimilar, so we are mutated as well, but we don't tell that we are carrying a disease. Okay, so mutation does not cause disease always. Now there are mainly two types of mutations. That is the point mutation, where the mutation takes place in a single base pair and it is being replaced by another base pair. One single base pair in a genetic code actually gets changed. Like we can give an example, like in case of sickle cell anemia, a point mutation in the beta globin chain results in the formation of valine from glutamate. Okay, in place of glutamate, valine is being replaced by it, and this causes the RBCs to change their shape and form into sickle shape. Okay, the second type of mutation is the frame shift mutation, where the change is in a reading frame due to insertion of or deletion of a total base pair. Okay, first we go through the insertion.
where a extra or more one or more nucleotides are being added in the DNA segment. Okay, if the insertion of three or multiple bases do not change the reading frame, and but they can add a new amino acid as well. That in case of deletion, there is removal of one or more nucleotides from the DNA segment. The deletion of the three or its multiple bases actually do not change the reading frame, but it actually removes one or more amino acid. Okay, this was about the types, mutation and its different types. Now, if we come to the structure of the tRNA molecule, which is the transfer RNA, it is also called the adapter molecule, and the structure was proposed by Francis Crick. Okay, and this adapter molecule could read the code on one end, and on the other end would bind to particular amino acids. Now, tRNA before genetic code was before the genetic code was invented, the tRNA was previously called sRNA, which was soluble RNA, and its role as an adapter molecule was later reported. Now, coming to the structure of tRNA, this is a secondary structure of structure. Okay, so it is like a clover leaf, but this is a 2D structure, so it is a secondary structure, but the three-dimensional tertiary structure depicts that it is a compact inverted L-shaped molecule, the structure of tRNA. Okay, now the tRNA molecule actually have 5 parts. The first one is the anticodon loop. The bottom one is the anticodon loop, where you can find the codons UCA or AUG, which has complementary bases to the code present on the mRNA. Okay, the second part is the amino acid receptor end, which actually this is the opposite end of the anticodon loop, which actually binds the amino acid. The third is the T loop, which helps in binding the ribosome, and fourth is the D loop, which helps in binding the amino acyl synthetase, the enzyme required for translation, and the fifth one is a small outgrowth, which is known as the variable loop. Now, each tRNA is obviously specific for a particular amino acid, and a specific tRNA for initiation is actually called a initiator tRNA. But there are no tRNAs for any kind of stop codons. Okay. Now, today up to this, we will continue with the next part of the chapter in my next videos. Thank you.